Assalamu alaikum and welcome everyone to our molecular biology ASCP preparation series. This program is all about providing real life MCQs for your certification success. The MCQs we'll discuss today and in the upcoming season have been carefully collected from several years of past exams to give you the best preparation possible. I'm Dr. Loya Abdelati, certified as international technologist in molecular biology, and I'm really excited to share my knowledge and experiences with all of you through these series. So we are starting with the second part of molecular science, and our focus will be on the basic molecular theory. So let's dive in together, but don't forget to subscribe to our channel to receive all the latest lecture. I also encourage everyone taking the test to share your question with us so that everyone can be benefit. All right, are you ready, guys? So let's get started. All right, the molecular science part is divided into four sections: nucleic acid chemistry, basic molecular theory, biochemical reagents, and genetics, which together make up 20 to 25% of the total molecular ASCP exam. Actually, in the past two lectures, we completed the multiple choice question related to the nucleic acid chemistry. Now we are moving on to the basic molecular theory sections where all the concepts will be tested through the MCQs. All right. Actually, for those who missed the previous two lectures on the nucleic acid chemistry, I have added the links to those lectures in the bio below for your reference. So do not miss it, and don't miss to subscribe, to share and like our channel to receive all the latest lecture. Okay, let's move on. All right, now let's focus on the question about replication. I will begin by focusing on replication in eukaryotic cell and review ASCP questions from the previous exams. Are you ready? Let's go and see the first question in this group. Okay, here is the first question we have for today. DNA replication in eukaryotics begin at the specific site called 10 seconds. All right. Origin of replication. The correct answer is origin of replication. Origin of replication are specific DNA sequences where replication begins. All right. So let's move to the next question. The next question is, which direction does DNA synthesis occur during replication? Actually, DNA polymerase can only add nucleotides to the 3' end of growing DNA strand. So, synthesis occurs in the 5' to 3' direction. All right? So, the correct answer is B. So, let's move on. What distinguishes the leading strand from the lagging strand during replication? 10 seconds. All right. The leading strand is sensitized continuously in the 5' to 3' direction, while lagging strand is sensitized discontinuously at the Okazaki fragments. All right, let's move on. Let's take a look at the next question. And the question is, why is the DNA replication described as the semi-conservative? All right, let's move and see the correct answer. Actually, in semi-conservative replication, each daughter DNA molecule consists of one parental strand and one newly sensitized strand. Okay? This is why DNA replication is referred to as semi-conservative. So the correct answer is each new DNA molecule consists of one old and one new strand. All right? Let's move on. The question is, during which phase of the cell cycle does DNA replication occur? 10 seconds. All right. Actually, DNA replication occurs during the S or synthesis phase of cell cycle. This phase is dedicated to doubling the DNA content to ensure that each daughter cell receive a complete set of genetic information during cell division. All right? All right, the next question is, during which phase of the cell cycle does the cell grow and prepare for DNA replication? Think, think. 10 seconds. All right. The correct answer is G1 phase. G1 phase or gap 1 is the phase where the cell grows, sensitizes RNA and proteins and prepare for DNA replication. All right. So let's move to the next question. Which of the following occur in the G2 phase of the cell cycle? 10 seconds. All right, let's move on. In the G2 phase, the cell sensitizes protein needed for mitosis, grows further, and repair any DNA damage from the S phase. So the correct answer is preparation for mitosis. Okay? All right, the next question is, what is the main event during the M phase of cell cycle? 10 seconds. All right. All right. The M phase is the short for mitotic phase. Is when the cell will get busy. First, there is a mitosis where the nucleus is split, I mean nuclear division. Then comes cytokinesis where the cytoplasm divides. Boom, then you get two daughter cell ready to roll. So the correct answer is the chromosome separation and cell division. That is the main event during the M phase. All right, let's go to the next question. Okay, the question here say, which phase is also known as the resting phase where the cell exit the cell cycle and stop dividing? 10 seconds. All right. The G0 phase is the quiescent state where the cell exits the cycle and doesn't divide it, often seen in non-dividing or differentiated cells. Okay, so the correct answer is G0 phase. Let's move to the next question. What is the primary checkpoints before the cell enters the S phase? 10 seconds. All right, let's move on. Actually, the G1S checkpoint ensures that the cell is ready for DNA replication by checking for DNA damage and ensuring sufficient resources for division. All right, so the correct answer is G1S checkpoint. So let's move to the next question. The question is, which checkpoint in the cell cycles ensures the DNA replication is completed before mitosis begins? 10 seconds. All right, let's move to see the correct answer. The G2M checkpoint ensures that DNA replication is fully completed and free of error before mitosis. So the correct answer is G2M checkpoint. All right, let's move on. So the question is, during which phase of the cell cycles do the replication origins become licensed to the begin DNA replication? 10 seconds. Focus, focus, focus. All right, let's move and see the correct answer. In the G1 phase, the replication origin are licensed by loading the pre-replication complex. So during early S phase, replication begin at this license origin. All right. So the correct answer is G1 and early S phase. All right. Let's move on. During the G2M checkpoint, the cell verifies that. All right. As I said, G2M checkpoint ensures that the DNA replication is complete and checks for any damage before mitosis begin. So the correct answer is DNA damage has been repaired during the G2M checkpoint. All right. I hope it's clear. Now let's move to the next question. What is the purpose of the G2 phase following DNA replication? All right, let's move on. Okay, as I said, in the G2 phase ensures that the DNA integrity and prepare the cell for division by checking for error. So the correct answer is to check for DNA replication error and prepare for mitosis. All right, let's move on. So the next question is, in which phase do chromosomes align at the center of the cell? Let's move and see. During the metaphase part of the M phase, okay? Chromosome align along the metaphase plate, preparing for separation. All right, let's move on. What happens during the anaphase of mitosis? All right, let's move. In anaphase, spindle fibers pull sister chromatids apart to opposite ends of the cell to ensure each daughter cell receive identical set of chromosome. All right, so the correct answer is sister chromatid are pulled to opposite poles. Okay, let's move on. The next question is which phase involve the division of the cytoplasm result in two daughter cell? All right, cytokinesis. Cytokinesis is the process where the cytoplasm is divided into two. Yes, correct, two, completing cell division and forming two identical daughter cells. Okay, let's move on. Okay, the next question is how many origins of replication are typically found on eukaryotic chromosome? All right, let's move on. Actually, in eukaryotic chromosome have multiple origin of replication. Why? To speed up the process due to their large size of the chromosome. So the correct answer is multiple origin distributed along the chromosome. All right, let's go. The next question is what would happen if DNA replication didn't occur before cell division? All right. Of course, without DNA replication, daughter cells would receive only a portion of the parent cell's DNA instead of complete set, right? Leading to what? Leading to incomplete or missing genetic structure. This would prevent the cell from the function properly, and this can be also lead to cell death or dysfunction or genomic instability. Okay, so let's move on to the next question. Why is the replication more complex in eukaryotic cell than prokaryotic cells? All right. Because of eukaryotic DNA is linear and much larger, requiring multiple origins of replication. So the correct answer is eukaryotic cell have multiple origin of replication and linear DNA. Okay. Here is the question. What happens to the newly sensitized DNA strands after replications? Okay, let's. So after DNA replication, the newly sensitized DNA strands are wrapped around histone protein to form nucleosomes. Okay, so these nucleosomes further coils and fold to organize the DNA into chromatin, a compact structure that fits within the cell nucleus. Okay, this packaging is essential for DNA stability, protection, and regulation of gene expression. So the correct answer is they are packed into nucleosomes and organized into chromatin. All right. Okay, the next question is what is the result of errors that occur during the DNA replication and are not corrected? All right. The correct answer is mutation. Mutation may occur in DNA sequence as unrepaired errors in DNA replication can result in mutation. Okay, let's move on. Here, the question is which DNA sequence feature is the most abundant at eukaryotic origins of replication? All right. The correct answer is AT-rich region, as AT-rich regions are easier to unwind because of adenine thymine base pairs have only two hydrogen bonds. All right. So the correct answer is AT-rich region. Okay, the next question is which mechanism corrects replication errors immediately after they occur? Okay, let's move the result. The correct answer is proofreading by DNA polymerase. Actually, DNA polymerase had the proofreading ability to identify and correct errors during synthesis. All right, let's move on. What would happen if single strand binding protein were absent during replication? Okay, let's move and see the result. Okay, the correct answer is the DNA strand would reanneal, stopping replication. Without a single strand binding protein, single strand DNA would be reanneal, preventing the replication machinery from the progressing. All right, let's move to the next. Okay, the next question is how does the chromatin structure affect replication in eukaryotes? 10 seconds. All right. Actually, the chromatin is tightly packed in the form of nucleosome, right? Which can make DNA less accessible to the replication machinery. So, before replication can occur, chromatin must be partially unwrapped to allow the replication machinery to access the DNA. So, the correct answer is tightly packed chromatin slow down replications. All right. Let's move to the next question. All right. What is the significance of bidirectional replication in eukaryotic DNA? 10 seconds. All right. Let's move. Actually, the significance of bidirectional replication is to speed up the DNA synthesis process by progressing in both directions from the origins. This allowing for faster and more efficient replication. So the correct answer is option C. Okay, let's see the last question we have today. Which strand of DNA is sensitized continuously during DNA replication? 10 seconds. All right, let's move on. Okay, the correct answer is the leading strand. The leading strand is sensitized continuously in the 5' to 3' direction toward the replication fork. All right, this is the last question for today. Thank you for your attention, and I will see you in the next lecture. But before that, don't forget to subscribe to our channel to get the latest lecture. That's all for today. Salam.